Welcome students. Last episodes we discussed the structure of DNA and various forms of DNA, and then how the nucleic acid structure is stabilized, and then genome organization of prokaryotes and eukaryotes. Up to this, first unit is completed. In the second unit, we discussed the different enzymes involved in the DNA replication and how the DNA is replicated and what are the different replication method of DNA, and then how the replication is stabilized or standardized, or and the fidelity of the DNA replication with these some extra-chromosomal replicons, and then how the DNA is damaged during the replication and how it is automatically repaired during the replication. These are the topics which are discussed in the second unit in 4 different episodes. In this episode, we discuss what are the enzymes which are involved in the DNA replication. As all we know, a fundamental property of living organism is their ability to reproduce. Organisms produce offspring that are similar to themselves. The process occurs by which a single parent cell divides to produce 2 identical daughter cells. In this process, the genetic material must be duplicated prior to cell division so that the daughter cells receive a full complement of genetic information. Replication means synthesis of daughter nucleic acid molecules identical to the parent nucleic acid. Thus, accurate and complete replication of DNA is essential to the ability of a cell organism to reproduce. It is the basis for biological inheritance. DNA is made up of double helix of 2 complementary strands. During replication, the strands are separated. There are many enzymes. They participate in the process of replication. So, what are the enzymes which are involved in the DNA replication? The enzymes involved in DNA replication are DNA primase, DNA gyrase, DNA helicases, DNA clamp, DNA ligase, topoisomerases, DNA polymerases, RNA primer, a restriction enzyme, exonucleases, telomerases, and single strand binding proteins. First, the DNA synthesis starts with the starting point, which is activated by the primer. So what is meant by a primer? A primer is a strand of nucleic acid that serves as a starting point for DNA synthesis. It required for DNA replication because the enzyme that catalyzes this process. DNA polymerases can only add new nucleotides to an existing strand of DNA. The polymerases starts replication at the 3' end of the primer and copies the opposite strand. In most cases of natural DNA replication, the primer for DNA synthesis and replication in a short strand of RNA. The first enzyme is DNA primase. DNA primase is an enzyme involved in the replication of DNA. DNA primase is in fact a type of RNA polymerase which creates an RNA primer. DNA polymerases uses the RNA primer to replicate single-strand DNA. Primase catalyzes the synthesis of short RNA segment called a primer complementary to a single-strand DNA template. Primase is a key importance in DNA replication because no known DNA polymerases can initiate the synthesis of DNA strand without an initial RNA or DNA primer. The next one is chromosome. Chromosome is a complex protein containing chromosome A, chromosome B, chromosome C, DNA T, DNA B, DNA C, and primase protein, that is DNA G. Chromosome complex without primase referred to as perichromosome complex. Chromosome complex play a vital role in lagging strand synthesis by synthesizing the primer at frequent intervals. The primase A, primase B, primase C necessary for the binding of chromosome complex to DNA. DNA B responsible for helicase activity. DNA C aids the binding of DNA B. DNA T helps the binding of DNA B and DNA C complex to primase A, B and C complex. The next enzyme is DNA gyrase. DNA gyrase, often referred to simply as gyrase, is an enzyme that relieves strain which double-strand DNA is being unwound by helicase. This causes negative supercoiling of the DNA. The ability of the gyrase to relax positive supercoil comes into play during DNA replication. The right-handed nature of the DNA double helix causes positive supercoils to accumulate ahead of translocation enzyme. In the case of DNA replication, a DNA polymerase. The ability of gyrase, that is topoisomerase 4, to relax positive supercoils allows superhelical tension ahead of polymerase to be released so that replication can continue. Negative supercoiling of bacterial DNA by DNA gyrase influences all metabolic process involving DNA and is essential for replication. Gyrase supercoils DNA by mechanism called sign inversion, whereby a positive supercoil is directly inverted to a negative one by passing a DNA segment through a transit double-strand break. Gyrase is a prototype for a growing class of prokaryotic and eukaryotic topoisomerases that interconvert complex forms by way of transit double-stranded breaks. The next enzyme is DNA helicases. DNA helicases are often used for separate DNA strands of double-stranded helix or a self-annealed RNA molecule using the energy from ATP hydrolysis, a process characterized by the breaking of hydrogen bond between annealed nucleotide base. They also function to remove nucleic acid associated protein and catalyze homologous DNA recombination. They are proteins which are involved in the unwinding of DNA molecule. There are 4 kinds of helicases, namely DNA B, rep proteins, helicases 2 and DNA A. DNA A protein, which have the molecular weight of 48,000 kilodalton. It binds to 4 of 9 MER sequence and unwinds a 3 of 13 MER sequence at origin of C site and forms an open complex during initiation of replication. It is the first protein which binds to DNA to initiate DNA replication. The next complex, DNA B protein, with the molecular weight of 3 lakh kilodalton. It is a primosome constitute and consists of 6 subunits. It unwinds DNA during replication. It is responsible for the extension of open complex during replication. Rep proteins with the molecular weight of 65,000 kilodalton. It has a helicase consisting of one subunit. It binds to the 5-3- template and moves in 3-5- direction. It actively participates in leading strand synthesis in replication. The next one is DNA helicases 2 with the molecular weight of 75,000 kilodalton. It is helicase consisting of only one subunit. It binds with 3-5- template strand and moves along in 5-3- direction. It is involved in lagging strand synthesis in replication. The role of helicases is to unwind the duplex DNA in order to provide a single stranded DNA for replication, transcription and recombination, for instance. The next one is a DNA clamp. A DNA clamp is also known as sliding clamp, is a protein fold that serves as a processability promoting factor in DNA replication. As a critical component of the DNA polymerase 3 holoenzyme, the clamp protein binds DNA polymerases and prevents this enzyme from dislocating from the template DNA strand. The clamp polymerase protein-protein interactions are stronger and more specific than the direct interactions between the polymerase and the template DNA strand. The next enzyme is DNA ligase. The DNA ligase are responsible for connecting DNA segments during replication, repair and recombination. They are class of enzymes that catalyze the formation of alpha phosphodiester bond between two DNA chains. These enzymes recovers the free hydroxyl group at the 3' end of the other DNA strand and phosphate group at 5' end of the other. The formation of phosphodiester bond between this group is an androgenic reaction, hence energy source required for ligation. In E. coli and the other bacteria, NAD+ supplies the energy, whereas in animals, that is in eukaryotics, ATPs play the role. It plays a role in repairing single-stranded breaks in duplex DNA in living organism, but some forms such as DNA ligase 4 may specifically repair double-strand breaks, that is, break in both complementary strand of DNA. The ligase reaction which proceeds in 3 steps. First one is adenylation, that is addition of AMP of a lysine residue in the active center of the enzyme. Pyrophosphate is released. Second one is transfer of AMP to the 5'-phosphate of the so-called donor, formation of pyrophosphate bond. Then third step is formation of phosphodiester bond between the 5'-phosphate of the donor and the 3'-hydroxyl of the acceptor. In mammals, there are 4 specific type of ligase, that is DNA ligase 1, DNA ligase 2, DNA ligase 3 and DNA ligase 4. DNA ligase 1 ligates the nascent DNA of the lagging strand after the DNA polymerase 1 has removed the RNA primer from the Okazaki fragments. DNA2 ligase, alternatively spliced form of DNA ligase 3, found in non-dividing cells. DNA ligase 3 is the complex with DNA repair protein XRCC1 to aid in sealing DNA during the process of nucleotide excision repair and recombinant fragments. DNA ligase 4 is a complex with XRCC4. It catalyzes the first step in the non-homologous end joining DNA double-strand break repair pathway. It is also required for recombination, the process that generates diversity in immunoglobulin and T-cell receptor loci during immune system development. The next enzyme is topoisomerases. Topoisomerases are of group of enzymes which control supercoiling of DNA, thereby maintaining it in the proper topological state or superhelical tension. There are 2 classes of topoisomerases. They are type 1 topoisomerase and type 2 topoisomerases. Type 1 topoisomerase. Type 1 topoisomerase are nicking closing enzyme or monomeric 100 kilodalton proteins that are widespread in both prokaryote and eukaryote. They can remove negative supercoils without leaving nicks in the DNA molecule. The mechanism behind the type 1 topoisomerase activity is after the enzyme binds to a DNA molecule and cuts one strand, the free 5'-phosphate of the DNA is covalently attached to tyrosine residue in the enzyme. In the case of prokaryote, the free 3'-phosphate on the DNA is covalently attached in the case of eukaryotes. The DNA strand that has not been cleaved is then passed through the single strand break. The cleavage strand is then resealed. The type 1 topoisomerase from E. coli acts on negatively supercoiled molecules but not on positively supercoiled molecules. In contrast, type 1 topoisomerase from eukaryotic cell can remove the both positive and negative supercoils. The type 1 topoisomerase reversibly interlinks single strand circles. Type 2 topoisomerase. The first type 2 topoisomerase to be described was isolated from E. coli and named DNA gyrase, which have the 365 kilodalton proteins that consist of 2 pair of subunits designated as A and B. Type 2 topoisomerase enzyme have the ability to cut both strands of a double strand DNA and pass another portion of the duplex through the cut and reseal the cut in a process that utilizes ATP. Depending on the DNA substrate, these movements will have the effect of changing a positive supercoil into a negative supercoil or of increasing the number of negatively supercoils by 2. The type 2 topoisomerase enzymes from mammalian cells cannot, like E. coli DNA gyrase, which increase the superhelical density at the expense of ATP. Presumably, no such activity is required in eukaryotes since binding of histone increases the potential superhelicity. All type 2 topoisomerase catalyze catenation and decatenation, that is, the linking and unlinking of two different DNA duplex. DNA gyrase has the ability to cut a double-strand DNA molecule, pass another portion of DNA duplex through the cut and reseal the cut. It changes the linking number of DNA by 2. The next enzyme is DNA polymerase. The DNA polymerase enzymes copy DNA sequences by using one strand as a template. The reaction catalyzed by DNA polymerase is the addition of deoxyribonucleotides to a DNA chain by using dNTPs as substrate. All DNA polymerase require a template strand which is copied. DNA polymerase also require a primer which is complementary to the template. The reaction of DNA polymerases is thus better understood as the addition of nucleotide to a primer to make a sequence complementary to a template. The best studied bacterium E. coli has 3 DNA polymerase. DNA polymerase 1, which is primarily a repair enzyme, although it also has a function in replication. About 400 polymerase 1 molecule exist in a single bacterium. DNA polymerase 1 only makes an average of 20 phosphodiester bonds before dissociating from the template. These properties make good sense for an enzyme that is going to replace damaged DNA. DNA polymerase 1 has nucleolytic activities which are an intimate part of their function. The 3' to 5' exonuclease activity removes base pair sequence of the polymerizing activity. During replication, this can remove primers ahead of the polymerizing function of the polymerase. The another important function of DNA polymerase 1 is the 3' to 5' exonuclease activity. This activity can depolymerize DNA starting from the newly synthesized end. The 3' to 5' exonuclease activity serves an editing function to ensure the fidelity of replication. DNA polymerase 2. DNA polymerase 2 is a specialized repair enzyme like polymerase 1. A large number of polymerase 2 molecule residue in the cell is about 100. The enzyme is more processive than polymerase 1. Polymerase 2 has the same editing 3' to 5' activity as polymerase 1 but not the 5' to 3' exonuclease activity. The actual repair enzyme in E. coli is DNA polymerase 3. The property contrasts with polymerase 1 and 2 in several aspects. Polymerase 3 is much more processive than the other enzyme, making about 5 lakh phosphodiester bonds on the average. In other words, it is about 5000 times more processive than polymerase 1 and 50 times more processive than polymerase 2. Polymerase 3 enzyme is a multi-subunit enzyme. It lacks a 5' to 3' exonucleolytic activity, although a subunit of the enzyme carries out the editing, that is 3' to 5' function during replication. The polymerase 3 enzyme synthesizes DNA at least 100 times more rapidly than the other polymerases. It can synthesize half of the bacterial chromosome in a little more than 20 minutes, which is fastest than the bacterium can replicate. The actual replication enzyme in E. coli is DNA polymerase 3. Its property contrasts with polymerase 1 and polymerase 2 in several respects. The different properties of 3 different polymerase enzymes are comprised as the structural gene in the polymerase 1 is polymerase A and the 2 is polymerase B and then 3 is polymerase C. The molecular weight of polymerase 1 is 1,03,000 and then polymerase 2 is 90,000 and then polymerase 3 is 1,30,000. The number of molecules per cell for polymerase 1 is 400 and then polymerase 2 is 100 and then polymerase 3 is 10. The minimal initial rate, that is nucleotides per second, for the polymerase 1 is 16 to 20 nucleotides, whereas in polymerase 2 it is 2 to 5 nucleotides per second and then 250 to 1000 for polymerase 3. The 3' exonuclease activity is present in polymerase 1 and 2, whereas it is absent in polymerase polymerase 3. The 5' exonuclease activity is present only in the polymerase 1, whereas it is absent in both polymerase 2 and polymerase 3. Progression of the enzyme is 3 to 200 for polymerase 1 and 10,000 for polymerase 2, whereas it is 5 lakhs for polymerase 3. The biological role of polymerase 1 is DNA replication, DNA primer cleaving, and then polymerase 2 is SOS DNA repair, and then polymerase 3 is replicative elongation. In eukaryotes, there are 6 important DNA polymerase, which is referred as DNA polymerase alpha, beta, gamma, sigma, delta, and then epsilon. DNA polymerase alpha, which is composed of 4 subunits, of which one has the primase activity. The largest subunit of it has polymerase activity 1. It is supposed to exist in several forms like alpha 1, alpha 2, and alpha 3. DNA polymerase in association with DNA polymerase delta, DNA polymerase alpha. It is composed of 4 subunits, of which one has the primase activity. The largest subunit of it has polymerase activity. It is supposed to exist in several forms like alpha 1, alpha 2, and alpha 3. DNA polymerase alpha is association with DNA polymerase delta is involved in the replication of nucleic chromosomal DNA. The replication is also aided by another protein factor called accessory protein and accessory protein 4A. This protein factor appeared to have regulatory function. DNA polymerase alpha is supposed to carry out lagging strand synthesis. DNA polymerase beta, which is involved in the repair of DNA. DNA polymerase sigma involved in the replication of mitochondrial DNA. DNA polymerase delta has polymerase function and 3' to 5' exonuclease activity, which is involved in the leading strand synthesis. DNA polymerase epsilon is supposed to replace DNA polymerase delta in some situation such as DNA repair. DNA polymerase sigma seems to expressed only in the bone marrow cells. It is the only eukaryotic polymerase that has deoxyribonuclease activity. The next enzyme is RNA primers. RNA primers are single-strand oligonucleotide with 40 to 60 base pair. For replication, DNA primers are required. This is because DNA polymerase require RNA primers for their action to start in vivo condition. In replication, RNA primer is synthesized by 2 different enzymes, namely RNA polymerase and primosome complex. RNA polymerase synthesize RNA primer for synthesis of leading strand, whereas primosome synthesize RNA primer for lagging strand synthesis. In addition to the RNA polymerase synthesis, only one primer, whereas primosome synthesize many primers. The next enzyme is restriction enzyme, which is also referred as restriction endonuclease. This enzyme that cuts DNA at or near specific recognition nucleotide sequence known as restriction sites. Restriction enzymes are commonly classified into 3 types, which differ in their structure and where they cut the DNA substrate at their recognition site, or if the recognition and cleavage sites are separate from one another. To cut DNA, all restriction enzymes make 2 incisions, one through each sugar phosphate backbone of the double DNA helix. These enzymes are found in bacteria and archaea and provide a defense mechanism against invading viruses. Inside a prokaryote, the restriction enzyme selectively cut upon foreign DNA in a process called restriction, while host DNA is protected by modification enzyme, that is a methylase enzyme. This modify the prokaryotic DNA and block cleavage. Together these 2 processes from the restriction modification system. The next enzyme is exonuclease. Exonucleases are enzymes that work by cleaving nucleotides one at a time from the end of the polynucleotide chain, that is outside of the polynucleotide chain. A hydrolyzing reaction that breaks phosphodiester bond at either the 3' or the 5' end occurs. It close relative in the endonuclease, which cleaves phosphodiester bond in the middle of the polynucleotide chain. Eukaryote and prokaryotes have 3 types of exonuclease involved in the normal turnover of mRNA. 5' to 3' exonuclease, which is a dependent decapping protein, 3' to 5' exonuclease, an independent protein, and polymerase A specific specific 3'2 5' exonuclease. The next one is telomerase. Telomerase, also called telomere terminal transferase, is a ribonucleoprotein that is an enzyme that add DNA sequence repeats to the 3' end of the DNA strand in the telomere region, which are found at the end of the eukaryotic chromosomes. This region of repeated nucleotide called telomeres contains non-coding DNA and hinders the loss of important DNA from chromosome N. As a result, every time the chromosome is copied, only 100 to 200 nucleotides are lost, which causes no damage to the organism's DNA. Telomeres is a reverse transcriptase that carries its own RNA molecule, which is used as a template when it elongates telomeres, which are shortened after each replication cycle. The next one is single stranded binding protein. This single stranded binding protein binds to single stranded region of DNA to prevent premature annealing, to protect the single stranded DNA from being digested by nucleases, and to remove secondary structure from the DNA to allow other enzymes to function effectively upon. It was called as DNA unwinding protein with the molecular weight of 22,000 kilodalton. Single stranded DNA is produced during all aspects of DNA metabolism, replication, recombination, and repair. It contains 4 subunits. The major function of single stranded binding protein is to prevent recoiling of DNA strands after it unwinds by helicases. Thus, single stranded binding protein play a vital role in the replication. These are about the enzymes which are involved in the DNA replication. In comprising this episode, the enzyme primase provides an starting point of RNA for DNA polymerase to begin synthesis of the new DNA strand. DNA gyrase enzyme relieves strain of unwinding by DNA helicases. This is a specific type of topoisomerase. The enzyme DNA helicase, also known as helix destabilizing enzyme, which unwinds the DNA double helix at the replication fork. The DNA clamp is a protein which prevents DNA polymerase 3 from disassociating from the DNA parent strand. DNA ligase is the enzyme which reanneals the semi-conservative strand and join Okazaki fragments of the lagging strand. Topoisomerases are enzymes which relax the DNA from its supercoiling nature. DNA polymerase are the enzymes built a new duplex DNA strand by adding nucleotides in the 5' to 3' direction, and also perform proof reading and error correction. Telomeres are the enzymes lengthens telomeric DNA by adding repetitive nucleotide sequences to the end of the eukaryotic chromosome. And the last one is single stranded binding protein. Is a protein which binds to single strand DNA and prevents the DNA double helix from reannealing after DNA helix unwinds it, thus maintaining the strand separation. Thus we concluded this episode, and we will discuss new topic in the next episode, that is DNA replication and various types of DNA replication, and how the DNA is replicated in prokaryotes and eukaryotes. Thank you.